work on asymmetric cell division and how cells go from one cell to two cells. When that goes wrong, that becomes a cancerous cell. Divide out of control and have too many nuclei. If cells divide out of control, it becomes cancer cell and there's the tumor, right? So that's a problem for human disease. The other thing is that stem cells arise from asymmetric cell division. So my lab studies the bare basics of how that works. I have been working on C. elegans since I was a freshman in college. C. elegans is a really good organism to study embryonic development. I do imaging as well, and the embryo is quite big and transparent. And so we can see all the events that occur inside. I'm always excited about challenges. I teach this semester, so I think one thing that makes me really excited when students discover something new, and I love when I see them do that in class and also in my lab. That makes me really excited and also happy. I like mentoring students. I came from a family of artists, so I knew nothing about science. I knew nothing about how to be sterile. So I grew up with a lot of clay, crap on the floor of my house. In my house, there was no difference between science and art. I like the creativity in science, that you can, just like art, you can come up with an idea and then test it. Same thing you do in art. So you come up with an idea or a design or something, and you draw it or something and you create things. I do imaging, I do microscopy, and so for me, this art, you know, it's beauty inside the cell. It's like looking at a museum inside of a cell. I was really excited about being in a job where I can be really creative. No, you combine everything. So I have a food blog outside of labs. I make scientific cakes, so I combine everything I do. It's sort of seamless. You know, I do all of these things together. As long as you have a passion, you can find a way to combine them. You know, down in the foyer with all the images on the wall? Mm-hmm. So I did that. So my passion is also telling the public about science through art. So this is a way I've been able to use my art through outreach and getting people excited about science through the art. Right? And the artists see the science work and go, oh my gosh, they get inspired. So I collaborate with artists. I have artists that come to my laboratories. We give a talk, and I have people who come up to me who, if they have balls to come talk to me, then I always go, oh, and they're really interested. So that tells me that they're passionate. If that person comes to me and they're excited, I'm excited too. I highly recommend students coming in their freshman year, if not at the beginning of their sophomore year. Start early, and most faculty like to have people start really early. I give them their own experiments. Depending on how much time they have, they can act just like graduate students if they want. You got to start somewhere. I didn't know what the heck a worm was when I first started. I would open up a scientific bakery across the street. I'd rather bake scientific cakes than probably write grants. My other dream is actually to have a scientific art museum in Madison. It's really important to do outreach. Getting young women and underrepresented students into the science is really important, because discovery is made not just from everyone thinking the same. It's discoveries are made with a really diverse population. 3 different lasers, and they excite different colors. You know about GFP? So we put jellyfish gene into those worms that makes them glow in the dark. Sort of like a rave party, you know. So we make the worms rave on here. So we turn out the lights, and then the laser excites this. And we look at it live, and the embryos divide. So these are embryos developing with the GFP, you see? It localizes this in the cell cleaving there. See why you come to work every day? Well, what's happening here is all these 2 cells are becoming the brain, for example, of the worm. This one becomes your intestine, and this one becomes like their ovaries. So we know what each cell does. So every single cell from 1 cell to adult has been determined what it does.
I chose her lab because she does a lot of variety of research. So there's imaging and a lot of molecular work as well. I did a summer program here at the University of Wisconsin, my junior year summer. So I went to undergrad at St. Olaf College in Minnesota, and then I did a summer program here. But that's all the research experience I had before. So I study the role of a protein called ataxin-2 in cell division. Definitely do it. I think it's extremely important to get experience in a lab, otherwise you have no idea if you like it. In terms of getting into a lab, just be very persistent, email a lot of professors, show interest. I like the challenge, I like the logical aspect of it, having a question and trying to solve it.
I dream about it every day.